Great pleasure to be here. I have probably too many slides, so I will go fast, but it's recorded, so we can all go back. Okay, let's see here. So about 10 years ago, actually, we started realizing with the NGS data that there is something called computer sciences, and we're not experts in it. So dealing with these VCF files and sort of larger data sets can become quite a challenge for us and physician scientists. And we early on thought there must be a better way to do this. Initially, like many of you probably used Excel sheets, and after a while you can't even control which Excel sheets you saved where anymore. So these issues that come when you scale up, not just NGS for a family, but now you have maybe dozens or hundreds. We laid this all out already in 2013 in a paper here. It's really, how to manage these large data sets, especially when you don't have the resources. But how can you still maintain hands-on analysis as a non-bioinformatician? I think this is key, very key. And finally, as we all know now, of course, if we could find ways to combine these small sets of ataxia patients here and there from around the world, it can become a very powerful set of data. And that's really the promise. If we as a group succeed in this, we will maintain a high pace of discovery, which is key for our patients. We'll build scientific communities, which is also a lot of fun. And we really prepare seriously for translation of this work into clinical trials. So here's some of the papers we had published over the years, laying out these concepts. We were one of the founding members, for instance, for the Matchmaker Exchange. One of the first meetings was held here in Miami Beach. This is a sort of quick snapshot back actually to late 2012. All the major elements were already here on this, we at the time called GemApp platform, which is now Genesis. So this work has really led to great stories, great discoveries. Like here, this disarm gene is a major gene. Actually, 5 days ago, I just learned today, Lilly bought for over $100 million a company that was exclusively founded around this disarm gene. Unfortunately, I'm not a co-founder, so it didn't affect me. Another great example that you all know is the RFC1 repeat expansion. Andrea Cortesa here actually spent most of the time here in Miami working on this paper while we were sort of supporting Henry in this endeavor. And we have now established also in Genesis which is driven here by Sarah. And I can't tell you more about this today. I wouldn't dare, but there's potential for another major surprise discovery in the works actually, in ataxia and repeat expansions. So here's another example, great paper, just came out earlier this year with a cover story in Nature Genetics. And that is probably will lead to the very first treatable hereditary neuropathy. So something we also want to see for ataxias. So all in all, over the last decade or so, this has supported the discovery of over 80 Mendelian genes, and you can see it's a very diverse data set from around the world. It's really open to everyone. It's pretty much free to use. And here you see a bit of the content, some 12,000 cases right now in there. Most are exomes, but increasingly now about 10% are also whole genomes. And many different phenotypes. Probably you're pleased to see that ataxia is the #1 here, close to 2,000 rare ataxia exomes and genomes in Genesis right now. And equally exciting that a similar number of spastic paraplegias, pure and complex, and also peripheral neuropathies and others. Diseases that can sometimes overlap, so they can be very informative in analyzing these data. Here's an example how this typically works. Discovery of UBAP1 was really driven by a group in Iran that had samples data in Genesis, and they had this humongous family that pointed them to UBAP1 as a new gene. And then because of Genesis, we were quickly able to expand this finding more families, including data sets from Careful Rare, also Guy Rouleau, and of course, European samples. And as expected, since the Iranians were really driving force, they were leading this paper, and this is how it usually is done. And it's quite surprising in some of the fields where genesis investigators are very prominent, like peripheral neuropathies, that's myself. We have really started to dominate sort of, contributions in new genetics, new genes over the last 10 years or so. This is not just sort of the Miami group. This is really the concerted effort of investigators who use this tool, and they come from around the world. So the majority, so all the reds were sort of genes are discovered by, or supported by genesis. And we see sort of similar trends now with Ataxia and the PREPARE consortium, where over the last few years, since we do this, there is a good number of new genes or new genotype-phenotype correlations that are being have been described. There are several papers under review, novel genes also, and even more genes are being sort of prepared for publication. So this is great. I would say I've seen, sort of organizing a lot of the things in Genesis. I've seen a lot of different groups, and I'm really impressed by the Ataxia group, because this group here has agreed to share amongst each other openly, completely openly, all the data in real time. So everyone who submits even just a handful of data sets gets access to all the samples with Ataxia. And this slide in the background is not even complete. There are now a couple of investigators from South America who just joined, and also from Iran. I don't want to go too much into detail here. As you can expect, the back end always looks complicated for many of us, but as you would expect, the analysis includes the standard elements like SNV, structural variants, but also special things like repeat expansion analysis. And we have also an in-house developed AI prediction pipeline now, which beats everything that's been published in this area. There's some certain principles. We only take pseudonymized data to ensure regulations. Data is private or shared depending on whoever uploads it, and the data always belongs to who uploads it. It never belongs to University of Miami or anyone. If investigators leave, then data will be removed. And of course, we try to always use open typing and open tools for analysis. Great sensitivity, of course, especially in Europe about GDPR, but I can assure you, same anxiety about GDPR in the United States now. So we have really implemented the required protocols there. And here are just some of the elements that ensure security on the system. In fact, we were recently ranked or rated as with a score of A+ in terms of security. The system runs on AWS, which in Azure. AWS is the cloud platform from Amazon, and Azure is from Microsoft.
Stephan, you have 2 minutes maximum.
Yeah, keep going. And so just pointing this out. So the interface, we really like to keep it nice and clean and nice to use, because it shouldn't look like a student project, so to say. And I'm just giving you some examples here, including this is how you share data. And at the end, really, I also want to point out, we have published, my group has published on, for instance, burden analysis in CMTs. This would not have been possible without the work we have done in Genesis. So this is sort of a secondary result from this. And the same is for modifier studies, like here's an example. And the very last slide, Mathis, it's very important how to pay for all of this, because to do this, you need expensive servers, expensive personnel. And the key thing here is we get support from many different corners, including grants, but also the MDA has given us a special recognition grant recently to support it, and private gifts even. Thank you.
Thank you, Stephan. That was really nice. I'm just a little bit German harsh here because I want to keep some time for the roundtable at the end. So, Sergi, I think you're second. Can you take over 10 minutes sharp?
Yes.
So Sergi is leading the Solver-D network in Europe and has launched the GPad pipeline, which is one of the most prominent NGS pipelines in Europe.
Okay, thanks a lot, Mathis, for the invitation, and also to Stephan for the nice introduction of why we need platforms like the ones we are discussing today. So I can spare that part. I'll go directly into what the GPad is. Okay, there we go. Okay, so the GPad, we call it the Genome Phenome Analysis Platform, is an online system that facilitates collation, sharing, analysis, and interpretation of integrated genome phenome datasets for rare diseases, and we think that's usually for difficult cases to diagnose, but also for gene discovery. So in a very similar way than what Stephan showed. What we do is collect on one side the phenotypic data, and we use the standards for that, and on the other side we collect the genomic data. We collect the raw data, either a FASTQ or a BAM or a CRAM, because that way we can standardize the way that we process the data, and all the data is comparable within the system. It is now a NERTED recognized resource. I think it's been there for a couple of years now. We have a similar number as to what Stephan mentioned, about 13,000 genome phenome data sets. Not all of them are index cases. We also have relatives of the individuals affected. Data is pseudonymized in the system, and we ask that data becomes available to all the registered users within 6 months of submission. There are some exceptions that can be granted, but the idea is that we can all collaboratively work towards diagnosing those cases. And if you are not able to do it within 6 months, then our idea is to try to work together on cracking some of those. The registration for users and data access is regulated by a data access committee. We have a code of conduct and an adherence agreement that all the users need to sign and abide to or abide by. The activity of the users is logged, and also the security of the platform is audited periodically by an external company. So we are now moving into a new module for collecting all the phenotypic data. We are using until now Phenotypes, which is developed by Mike Brudner's team in Canada, and as part of EJPRD and a little bit with Solver-D, these are the two European projects, we have developed what we call Finnstore, and this is a system that allows us to integrate all this phenotypic and clinical data in a standardized way. So you can either go in and with a user-friendly interface, create your case and your family, your pedigree, or you can also import cases in a bulk way with different approaches, either using JSON or using PhenoPackage, for example. That is how one of these forms look like. You would go into each of these submenus, and then you would enter all this information with a click way. And what we do is create templates to facilitate entering this information for different disease groups. And what we have done now as part of Solver-D is create templates for all the major disease groups that were defined by Genomics England. They are publicly available on their website. And this is one of the cases how it would look for the human being. These are HPOs, and at the bottom we use orphan hereditary ontology and immune identifiers for disease classification. Obviously, the beauty of this is that we can use these codes, these ontologies, and they are machine readable, and then we can use them to do programmatic analysis of the data, not just analysis by human beings. Recently, we have incorporated PhenoPackets as one of the standards that we can use for transferring data, for sharing data between different parties or stakeholders. This we have implemented as part of Solver-D, and PhenoPackage is basically a computer readable format developed within GA4GH that allows the standardization and structure language of phenotypes and ages and all the phenotypic features, but also some genetic features of the individuals in a format that you can transfer and import and export from different applications. This is how the user interface looks like. We are going through a new redesign of the system, and hopefully that will become available at the beginning of next year. But currently what we have is basically a top window where you specify which samples you want to analyze. You specify which filters you want to apply to these samples or these experiments. And then once you run the query, at the bottom you get a list of the variants that meet this criteria. And for each of these variants, you have lots of information to interpret these, to interpret them. Basically, you have information through many links that appear in different places, like at the position level or the gene level. But also in the middle tab, you have many subtabs where you already have a lot of integrated information that is either indexed in the system or accessed through web services to other servers to gain more information about these variants. These are some of the features of the system. So we have standard filters and annotations for SMBs, indels, and CMBs. We have filtering by genes of interest. That's usually useful if you really want to focus on a set of genes, or expand this a little bit more if you phenotypes or reactions, but you're not exactly sure what gene you're targeting. And we have different ways of doing that, including genomic Zealand panel app or Mendelian.co. We have also integrated phenotype-driven strategy through Exomizer. We can filter by variants annotated in the CLIMA database. We are able to filter by regions of runs of homozygosity, which we also use to assess consanguinity, and that's very useful to maybe change the way that you are filtering a case, because in some cases you might think there was consanguinity and there was not there, or the other way around, and that lets you change how you approach that case. There's direct links to multiple resources. You can tag and share candidate variants using ACMG guidelines, which allows you to collaborate on the cases, but also see if someone else have tagged a specific variant in a gene in another case, and that allows you to find similar patients within the system. You can search for variants in genes across all the samples for which you have access in the system, and it's a collaborative environment. You can share data and queries between users. And also the system enables anonymized data discovery through Beacon and matchmaker exchange. Some of the features that we can highlight is, for example, the search across all, which allows you to search for a gene across all the samples for which you have access. And then you can specify any type of filters from less stringent to more stringent. And then you will have at the end, yes, I have some background. Mattis, are you talking to me?
You have 1 more minute to go, Sergi.
Okay, excellent, good. I'll be quick. And then you can identify if any other cases have been tagged with that on that gene, for example. We have a new system to specify cohorts. So select the cases that are similar to some of the metrics that we have in the system, some of the descriptions of the cases. In this case, for example, we search for all cases with rare hereditary ataxia or ataxia described by HPO and any of their lower terms in Ontology, and we found we have over 1,200 cases meeting these requirements in the system. And also a lot of these analysis can be done programmatically. This is something we are developing now, and it means that you not only have to analyze the data with a graphical user interface, but if you know what you want to do, you can make the queries and then programmatically get out results from the system. And this is also something we are applicating a lot in SolveRD, and we have already got some good results with this approach. And finally, Stephan already mentioned that, but we are also connected to Matchmaker Exchange, mainly to Phenome Central and Decipher, but we are expanding those connections. And that's how it looks like, the internal connection, external connection. And that's it. I'll leave it here also with the slide. Again, as Stephan said, you usually need quite a bit of funding to be able to sustain such platforms in the long term. So thanks a lot.
Thanks a lot, Sergi. Very impressive. Really two great platforms. So maybe, Brent, you can take over next and present us your NGS platform.
Hello, and thank you for the opportunity to speak today. My name is Brent Fogel from the University of California, Los Angeles in the Departments of Neurology and Human Genetics. I'm going to be talking to you today about the Centralized Ataxia Genomics Core, or CAGC. These are my disclosures, and I have none relevant to the topic of this presentation. So, first off, there's an important need to have centralized repositories for genomic information in ataxia. I'm going to go through a couple of those reasons right now. First of all, cerebellar ataxia itself is a highly heterogeneous condition, both clinically and genetically. approximately 50% of patients with presumed genetic ataxia still remained undiagnosed after genomic testing. Mutations in ataxia genes are often rare or private and isolated in single families. So in order to validate these, it can be very difficult to find other patients with similar mutations or even mutations in the same gene. The scope of mutation, prevalence, penetrance, expressivity is unknown really for a large number of very rare ataxia genes. And ultimately, a precise genetic diagnosis is essential to improve research and advance clinical trials, particularly with the advance of gene therapy and methods of gene replacement that are going to be coming. Now, the CAGC repository that we've developed is based on an open source platform initially developed by Intel with the Broad Institute and MIT and Harvard. And the platform's called Genomics DB. And it's designed for an array storage of genomic data. And so this is illustrated here on the figure on the upper right. And so essentially samples are arrayed in one axis, whereas positions across the entire genome are arrayed in the other. And so this allows searching to be done in a number of different ways. It can be done by sample across the genome. It can be done by a specific genomic position. It can be done by intervals across the genome, but in a very organized, systematic way. Genomics DB contains a array storage manager to allow for high efficiency searches. It has a C++ library for importing large genomic files, VCF files, and it scales with the number of samples. And so what I'm showing here is a figure from the paper describing Genomics DB that illustrates the import and read time of the genomic data as the sample number scales up. And even at high sample numbers, it can still be utilized highly efficiently. So the upper limit of samples that Genomics DB can hold is dependent on the local storage capacity. And at UCLA, where this is currently hosted, we currently, that upper limit is currently undefined. So at this point, we have no limit to the number of samples that we can store in this database. Now, in terms of the data that is collected on each individual sample, it's a bit extensive, and that is to allow us to have maximum flexibility for normalizing the data for analysis. I'm going to talk about that in a minute. But here's just an example of some of the key information that's obtained on each and every sequence that's imported into the repository. And it's a sequencing platform, library strategy, library source, the method of selection, the library layout, what instrument the data was sequenced on, what commercial kits were used, and all the information that'd be necessary to identify batch effects and such. And the reason for this is because there's no standardization of sequencing. And so depending upon the source of the sequencing, we could be receiving sequences from a wide variety of different methods. And in that case, we still need to normalize them so in order to be able to cross-compare across the data. And so this is an illustration of the pipeline that we use for data analysis. And I don't have time in this short presentation to go through exactly how it all works, but to illustrate the key features. Essentially, the sequence data is normalized as we just discussed, and then it's analyzed through a number of different pipelines. There's a classic pipeline for calling variants within the protein coding portion, so the exome or the protein coding portion of the genome. That's relatively standard. There's also modules for looking at copy number variation, so deletions and duplications that can be identified, as well as for repeat expansions, which are clearly an important source of genetic mutation and ataxic conditions. Now, one key aspect of this is it's optimized for continual analysis of the data in order to periodically reevaluate the different the sequence data in order to in case there's new information or new genes or new mutations are identified that could potentially be diagnostic. Now, this is what the user would see. This is a illustration of a standard user-initiated search. Essentially the initial information you need to submit is genomic position and the reference and alternate allele. So what we're looking at here is chromosomal position, a reference allele, the alternate allele, and the genomic position. This is illustrated for a classic mutation in the SPG7 gene. There's an advanced search that also allows input of a genomic region, and then future builds are going to allow submission of just a specific protein change and a gene name. So this is what the user search queue looks like. These searches are run continuously unless the user suspends them. And this is an illustration of 3 different searches for 3 classic mutations in recessive ataxia genes. If a match is made with any of these, the user receives a notification and they're provided with options on how to contact the other submitter or submitters in order to potentially collaborate. Now, this is an illustration of how this works in practice. This is a reanalysis of 76 patients who were originally exome sequenced and then reanalyzed 5 years later. And these are publications of ours that are shown here. And the key thing that I want to illustrate here is of these 76 patients, when they were reanalyzed, 24% of them were reclassified. So as additional information is added on new genes or on the pathogenicity or lack of pathogenicity of certain variants, it enables you to make more precise diagnostic opinions. And what's most significant about this is that 7% of the patients were reclassified as having pathogenic variants, which are ultimately diagnostic. And in 4% of those, more than half of them, it was a variant of uncertain significance that was reclassified as pathogenic. But in 3% of them, there was no significant variant identified the first time through, but a pathogenic variant identified on the reanalysis. So this is highly effective in improving diagnosis over time. Now, so again, this is the Centralized Ataxia Genomics Core. The website is listed here. It's currently accepting data. And again, just to hit the high points, this is a ataxia-specific data repository to confirm, validate rare genetic variants, and help to identify novel genes involved in ataxia. We intend for this to be a resource for innovative projects in investigating ataxia genetics and genomics. It's housed at UCLA, University of California, Los Angeles. We'll accept all types of sequencing data, exome or genome. And for institutions or anyone that considers genomic sequencing information to be protected health information, I just want to point out that our system is HIPAA compliant, which would basically allow for these sequences to be imported and stored while protecting patients' privacy, and that data is not, that information is not shared. So just to conclude, I'd like to thank the National Ataxia Foundation, who funded the development of this resource. I'd like to thank all of our patients who have contributed their data to it, ARCA Global for this opportunity to speak today, and I'd be happy to take any questions during the allotted period. Thank you very much.
Thank you very much, Brent. That was again really nice. So we have 7 minutes left. What I would like to use in those next 7 minutes, I would really like to discuss the question, how should we deal with those 3 different platforms? And I have prepared a slide to stimulate the discussion. But before bringing this up, I would just like to encourage all attendees to put questions or comments on those 3 platforms, which you have to one of those 3 speakers. Put that into the Q&A, and even though we will not have time to reply to this today in real time, we can try to copy those questions, and then we come back to you either tomorrow, or the speakers will come back to you by email. So please feel free to put your comments and questions in the Q&A. However, for now, I would like to use the last 6 minutes on the question. We have now seen those 3 nice NGS platforms. And what I would like to discuss with the 3 speakers is, how should we move on with SCA and ARCA Global in terms of which Ataxia NGS platform to take? And just to trigger off a discussion, I sketched out 3 different options, and some of them are maybe a little bit provocative, but this is just to get the discussion going and to start off from some options. Option number 1 would be each SCA and ARCA Global site should choose one favorite Ataxia NGS platform, so whatever the respective site likes, and then all 3 platforms focus primarily on API interfaces or matchmaking. That's option number 1. Option number 2 is, I called it the Liberty option. Each SCA or ARCA Global site chooses a favorite NGS platform, but then can also, for example, upload the respective data sets to the other platform. So that's just have a double upload. So that's, and then we don't have to worry a lot about matchmaking API interfaces. Option number 3 is we as SCA and ARCA Global should jointly prioritize one of those 3 NGS platforms, just by the pragmatic fact that the higher the numbers of unsolved Ataxia NGS datasets in one place, the higher the likelihood that we will be successful in identifying new genes. And for this, we might need to come up with criteria in order to select the one platform. For one criteria set might be to take that one with the most Ataxia NGS datasets and the most proven success of identifying Ataxia genes in the last years. So those might be criteria. So there's 3 different options just to start the round table. And I would like to hear your opinion. Which is your favorite option, or should it be a merged option out of 2 of those? What is your favorite way to go, and what would be your suggestion be for the Ataxia Global initiative?
Are we going in order? Stephan, do you want to go first? Or
Yes, Stephan,
I can
start.
Sure, I can start. I mean, basically, you know, different ways to skin a cat, as they say. I think honestly that this is not a. This should, in my personal opinion, this should not be one platform is sort of dominant over the other. I think there are different motivations why these platforms exist, who is behind it, slightly different philosophies, different technical abilities, and you name it, and so on and so forth. So I can tell you that we produced in Miami, we uploaded into the European tool, for instance, and I know there is data in Genesis that is also in the European tool. So, and these investigators have their reasons why they do it. So I think keep it open and let investigators and consortia make their decisions would be my personal. I would sort of try to not try to dominate, dictate how it's done.
Would that be a version of option number 2, the Liberty option?
Probably the libertarian. You see what I am, libertarian.
Okay. So it will not be political tonight. The US has a lot in the next 2 weeks to deal politically. No politics tonight. So Sergi, you want to take the next?
Yeah, no, I was actually gonna say that I would go for 1 or 3. So I would say, so I see the point what he's saying of the different abilities that the different platforms has, and I think it has been shown today that that's clearly that probably it should not be exclusive. Like if you put your data in one place, that should not prevent you from putting your data in another place, because you might lose some of the features that you might gain by doing that. On the other hand, the second option, what I don't like is that you might, as a global association, right, you might be losing the ability to identify all the data sets that are really close to each other and be able to maximize research by being able to put these data sets together. And that's why I think that, only option 1 or 3 gives you this ability, unless option 2 means that you put, or all of you put all your data in all the platforms, which I don't think is going to happen. So I think that there needs to be some level of interoperability or ways of querying data across systems to know, yes, look, we are working, I don't know, for example, with Genesis, and we know that we have 1,200 data sets there, but I know that, if I would expand to the other two, I would, be able to get access to 1,000 more, and at least be able to know that the data is there. That's the minimum that you can expect. And then if you can do analysis cross systems or not, that's another story. That's challenging. We all know that. But I think that's where a lot of us are going now. And definitely in Europe, that's where, for example, the 1+ million genomes is trying to go.
Thank you, Sergi. Brent, do you want to go next?
Sure. I actually agree with both points. I tend to favor option one. There again, as Stephan said, there's reasons for the different platforms to exist. There's different expertise in different sites, and merging them together prevents duplication and prevents reinventing the wheel. Like in our case, for example, our exomes that are done on our site actually can't be transferred onto these other platforms because of some of the institutional policies. So we wouldn't even be able to do option 3 even if we wanted to. So part of the value of option number 1 is if you're putting your data into any one of them, you're essentially searching all of them. That would really be the ideal for me.
But would that be technically feasible on a single variant level? So not just maybe sharing the results, but would you be, ideally, you would be able to query all data set at once, and that will most likely not be possible at all. So wouldn't a matchmaking or API model they will never really be really fruitful, efficient in daily life?
Brent has a good point. It is his institution, and I know other institutions have. There are some institutions who are quite strict. There are whole countries, take China, that are quite strict. And I bet you there's a platform in China that's probably happening now. And the day will come when we want to work with them also. So that alone tells you it can't be just all in one. Also, competition is good, a little bit of friendly competition is probably a good thing.
Maybe we can take it the other way around. Would anybody of you say we should not go for the Liberty option? Would anybody of you, Sergi, would you say we should not have data sets in Solvadi, GPub as well as Genesis? Would you be opposed to that?
No. So the option to the Liberty option, I don't see it incompatible with option 1, which is probably the one that we are saying that would work best, right? So yes, there is each site can choose where they put the data, but I think that we, and not only as as the ones presented today, but other systems also in the world need to work together towards this interoperability, and this gives the best of option 2 and option 1 together. But also option 1 is not going to be magically happening in a year or in 2 years. That's that's been work towards this for several years now, and Matchmaker Exchange is there. It has some. It works well, but it has some drawbacks also, and these need to be improved. And there are also other things that we cannot do yet with Matchmaker Exchange that we can do with a more, intrinsically federated system, like being able to do some of the queries that you were saying. I would like to be able to query Genesis and not only, look at the variants that someone has tagged. I would like to be able to query Genesis and say, I want to see if any of your patients has a variant on this gene of this kind or this kind, with these filters. And then, you run the query in your system and you give me the answer back. So I would like to be analysis on someone else's data without actually seeing the data. I think this is where we have to go at some point. But this is challenging, it's not straightforward.
At some point, I agree, Sergi, that's probably part of the future. As we both know, when you drill down to it, it's incredibly demanding technically to do that. And we have over the last, do this for a while now, we have learned, we had all versions of this. In the beginning, we could query all the variants, then you could do a gene-based matchmaking and automate it or through a person. And we come back actually back to person, meaning people contact my team and ask, hey, I have this candidate gene and can you look in Genesis? And this sounds like the most simplest, but I still, now I'm back to this because what I've learned from the automatic, when we made it automatic, people abused it. There's some postdoc somewhere, he's just lazy and runs 20 candidate genes that shoots off emails to all these clinicians. And after the 20th time, these clinicians say, I'm not going to answer to anything anymore. And this is also not what you want. So we have to be very respectful of the clinicians' patients with us. Because frankly, they expect things just work. They, you know.
This is how we clinicians are. So time is over. I think from here we have to take it in different steps. So I think what I get from the roundtable discussion is that nobody is really as opposed to putting up of uploading data into two different pipelines in parallel. So this will still be a possibility. Some centers will prioritize one of the platforms either because they like one more than the other, or they have the only institutional rights to do it in one, not as the others. Nevertheless, I think we have to find out the best ways to work either on technical APIs matchmaking, or whether we can find a good way of personal matchmakings without putting all the barriers on the clinicians. So I think those would be the next steps to do. I think those are for us to think about on the platform side is how can we work out the best solution on this? And for all of you as the SCA and ARCA Global partners, I think you have now gotten an overview on those three platforms. And I think for now, just please feel free to contact one of those three persons, Stephan, Sergi, or Brent. And I think we should start and get going with uploading your data sets, and then we just find a way to get this linked up together.